Hello, this is I.S. Kumar Farzana Banu, Assistant Professor in Botany, working in Maharani Science College for Women, Bangalore. In this video, I will explain a brief account of dormancy, which is prescribed in 3rd BSc Botany syllabus of Bangalore University. Let us walk through the PPT which I have prepared in this context. These are the contents of this topic. Let us move on to the introduction and definition of dormancy. Dormancy is a period in an organism's life cycle when growth and development and in animals physical activity are temporarily stopped. This minimizes the metabolic activity and therefore helps an organism to conserve energy. Dormancy tends to be closely associated with environmental conditions. In this image, you can see the seeds. So, during the developmental cycle of the plant, at some phase, certain structures like buds, tubers, seeds go through this period of temporary suspension of growth activity. Such a state is called dormancy. It is a derived dormant word. Dormancy is a physiological process in which growth is temporarily suspended. It is also called resting stage or dormant stage. So this is found in buds and seeds. When it is found in bud, it is called bud dormancy. When it is found in seed, it is called seed dormancy. So bud dormancy, when sprouting is temporarily suspended in a bud, it is called bud dormancy. Whereas seed dormancy, when germination is temporarily suspended in a seed, it is called seed dormancy. During the dormant period, the bud or the seed failed to germinate. Dormancy is an adaptation to unfavorable conditions. When favorable conditions resume, the normal activity starts and the bud's seeds begin to germinate. So coming to the bud dormancy. So as I had told already, the bud dormancy is adaptation where the sprouting of the bud or the germination is temporarily suspended. It is also called resting stage. So during this period, the bud remains inactive and it fails to sprout. So dormancy happens during unfavorable season. This is found in woody plants where the buds remain dormant during the winter season. Similarly, in case of potato, the buds, that is the eye spots, do not sprout immediately after harvest. They remain dormant for a considerable time. This period in potato and such other plants are called storage period. During bud dormancy, several morphological and physiological changes occurs. What are those? So that is extension of internode is checked, expansion of leaves is prevented. And here the apical meristem may continue its activity for some time and the bud size increases. But the meristematic activity stops after the formation of fully developed bud. So now coming to the causes of the bud dormancy. So the causes of the bud dormancy includes unfavorable season, short day length, phytochrome, so which is nothing but a protein pigment inducing flowering, and the fourth point is exhaustion of nutrients, increase in abscisic acid, decrease in gibberellins. So these are the causes. And the dormant bud resume to normal phase when favorable conditions arise. So now the resuming to normal phase from dormancy is called breaking of dormancy. So how to break bud dormancy? So these are the following factors. So the first factor is chilling treatment at 1 to 10 degrees Celsius for 10 to 40 days. Okay. So the second one is exposure to long day conditions. And the third one is chemical treatment with the thiourea, gibberellins, ethylene, chlorohydrin, dinitrophenol, etc. So this treatment can be given for the seedling stage of an plant. So coming to the seed dormancy. So what is this seed dormancy? The word dormant means sleeping. Seed dormancy is defined as a state in which the seeds are unable to germinate even under normal and favorable conditions for germination. Whereas this seed dormancy is a phenomenon where the germination is temporarily stopped or suspended in a seed. This seed is called now dormant seed. So this dormant seed is in a resting stage. During this stage, the seed failed to germinate. The most of the this particular thing can be seen in agricultural and horticultural plants usually that germinate promptly if that is given moisture, suitable range of temperature, etc. So there is, however, in more number of plants whose seeds do not readily germinate even though they are placed under favorable conditions of moisture, air, temperature and light. Germination may be delayed for weeks, the days, weeks or even months together. The seeds of such plants are said to be in a dormant condition. Now coming to the classification of seed dormancy. So the types of seed dormancy include primary dormancy. So under that subheading, that is physical dormancy, mechanical dormancy, chemical dormancy. The second type is secondary dormancy, under that physiological dormancy, and the lastly that is morphological dormancy. So now coming to the first one, that is primary dormancy. So this exogenous dormancy is a result of factors imposed from outside the embryo. For example, the seed coat and or the fruit parts which are hard. So this material surrounding the embryo may inhibit the inhibition of water or limit oxygen to the embryo or prevent inhibitor leaching or conversely supplying inhibitor to the embryo. So under that physical dormancy. So dormancy that is caused by an impermeable seed coat. It is the result of impermeable layer that develop during maturation and drying of seed or fruit. It prevents the seed from taking up water or gases. As a result, the seed is prevented from germination until the dormancy is broken. Generally, the physical dormancy is the result of one or more palisade layers in the fruit or seed coat. Then chemical dormancy. So that is mechanical dormancy. Mechanical dormancy occurs when seed coat or other covering are too hard to allow the embryo to expand during germination. Now chemical dormancy. The presence of inhibitors within the seed or seed coat can cause dormancy. For example, coriander seed pericarp contain coumarin and paddy hull contains abscisic acid. Tomato juice salicylic acid. Coming to the secondary dormancy. Physiological dormancy, wherein the physiological dormancy prevents embryo growth and seed germination until the chemical changes occur. Under that, there is photodormancy and thermodormancy. Photodormancy is nothing but a light sensitive affects germination of some seeds. These photoblastic seeds need a period of darkness or light to germinate. Whereas thermodormancy. Seeds sensitive to heat or cold. Some seeds germinate only at high temperature. Example, amaranthus. And next one is morphological dormancy. In morphological dormancy, the embryo is underdeveloped or undifferentiated. And now coming to the causes for seed dormancy. So, some of the important causes are due to the single or combination of many different factors. So, let us study what are those. First one is water impermeability. The seed coat of many plant species are completely impermeable to water. They are hard seed coat or waxy. They are having hard seed coat or waxy coating. This condition is very common in seeds belonging to the families Leguminosae, Malvaceae, Convolvulaceae, Solanaceae, etc. that can be seen in the next slide in the images. Here the germination failed to occur until the water penetrates through the seed coat. So in this image you can see the legume seeds which are having the hard seed coats and the cotton seeds, and so also the chili seeds and Ipomia seeds. So the second point is gas impermeability. The seed coats of certain seeds are impermeable to gases such as oxygen and carbon dioxide. Since oxygen is required for the respiratory activity in germinating seeds, the seeds fail to germinate here. And the third point is mechanical resistance of seed coat to the growth of embryo. The common seeds of the weeds are Amaranthus, Capsula, etc. that have such a hard, tough seed coat that it prevents any appreciable expansion of embryo. Thus, these seeds remain dormant. So, in this image you can see. The fourth point is rudimentary and poorly developed embryos. So, in many plant species like Fraxinus excelsior and Ginkgo biloba and also the members of Orchidaceae, the seed dormancy may be due to immature and rudimentary embryo. In such seeds, the embryo does not develop as rapidly as surrounding tissue. Thus, when the seeds are shed, they are still imperfectly developed due to incomplete embryo in these seeds. The fourth point is dormancy requiring for after ripening in dry storage. In some seeds like apple, peach, pear, cherry, etc., the embryos are completely developed in ripe seeds, but the seeds fail to germinate even when the environmental conditions for germination are favorable. So, the embryo of such seeds does not germinate even if the seed coat is removed. These seeds germinate only after a period of rest. This period is called period of after ripening. In this period, the seeds must be kept under dry storage. So, next coming to the sixth point, germination inhibitors. Seed dormancy is caused in some plants by the presence of certain germination inhibitors in the seeds. For example, abscisic acid, ferulic acid, coumarin, phthalates, dihydroacetic acid, etc. The next point is light. Certain seeds are sensitive to light. The light sensitive seeds are called photoblastic. Certain seeds germinate after exposure to light. These seeds are called positively photoblastic seeds. So this can be seen in example lettuce, tobacco, tomato, etc. So these seeds remain dormant in the dark. Certain other seeds such as Helloborus niger will remain dormant on exposure to light. In this case, the plants are called negatively photoblastic. So now coming to the breaking of seed dormancy. So how to break seed dormancy? Resuming to normal activity of seed dormancy is called breaking of seed dormancy. The seeds germinate on breaking dormancy. The following are the methods to break seed dormancy. What are those? Let us study one by one. First one is scarification. What is this scarification? So this is a treatment by which the hard seed coat, which is ruptured or weakened or softened to break dormancy. It includes scratching the seed coat mechanically. So that can be seen in the image. To modify the hard seed coat for easy permeability. So like you can also use sandpaper for that, which is effective for this purpose. So next point is water treatment. Generally seeds will be soaked in hot water for few seconds and then soaked for 24 to 28 hours in cold water. So which make the seed coat to get softened and wash off the inhibitors. The next point is acid treatment. Soaking the seeds for few minutes, that is 5 to 6 minutes in concentrated sulfuric acid or hydrochloric acid, that modify the hard seed coat, that is impermeable seed covering. At the end of the treatment period, the seeds are washed thoroughly with water, then the seed germination is done or sowing is done. Next one is seed stratification. In this image you can see that one. That is, the practice of chilling the seeds is adapted to break dormancy because some of the inhibitory substances can be destroyed by cold or hot treatment. The chilling of seeds by placing them in different layers of soil is called seed stratification. So this stratification can be done by the layers of seeds alternating with the peat moss, soil, sand, etc. or suitable material can be taken. This will keep the seed at low temperature. Like that, seed stratification can be done. That is seen in this image. And coming to the fifth one, that is chemical treatment. The dormancy caused by inhibitors can be removed by the use of certain chemicals. They are nothing but gibberellic acid, kinetins, auxins, nitrates, thiourea, ethylene. So next one is, last one is dry storage. Dry storage promotes the after ripening in certain seeds which are dormant when freshly harvested. Freshly harvested seeds of many annuals are herbaceous like cereals, millets, etc. that fail to germinate until after a period of dry storage. Thank you for listening my talk.